We have 3 lectures that really deal with T cell recognition and what T cells do. And I didn't add anything else. I thought there were going to be 8, and I just thought that, okay, we just need to get through these 7. And then what we're going to do tomorrow, and it's probably going to be here, because I need a big whiteboard, and there's not one in this building that I can find. These are little bitty ones over there.
They're too small? Okay.
Yeah, they're too small, and we're going to do anatomy of immune response, and that's bring about 4 different colored pens and a big piece of paper, ledger size or whatever, and we're going to do the whole thing, whole immune response here. But today, we'll, when we get through these 3 lectures, we will talk about if you have any questions. But first, are there any questions about the stuff, the lies that I told you this morning? Anything that's just burning?
Oh, I have a question.
Yes.
So IgM and IG, what else?
D.
D. Those have affinity for the same kind of molecules, but they function differently, right?
IgD doesn't have a whole lot. IgD is only found in the in the membrane. Never is going to be secreted and do anything particular. And it really only functions as part of the process of maturation of the B cell. And so we don't worry about that. When it comes to what does IgM do, its Fc portion has some effective functions like fixing complement that probably get into, well, I might have to pull something up just to show tomorrow, but some of the antibodies, when they bind their Fc, or they bind their antigen, they'll fix complement and start that whole cascade that is very good for opsonization and for destruction of pathogens. Some of them, like IgE, bind to mast cells. Yes?
Oh, does anyone have a T cell maturation double?
The slides or the handout?
The handout. Thank you so much.
So yes, the question is yes, they have different effector functions because they have different heavy chain. The constituted genes have slight differences in what they do. And the main things I want you, I don't even want you to even worry about IgD. I just want you to know that it's there, and there was that whole alternative splicing thing. It has to do with maturing the B cell in the bone marrow. I really hope that you all appreciate that the first antibody made is IgM, because it's the first constant region gene downstream of the variable region gene segments. And so it gets rearranged to, it's the closest one, we transcribe through IgM and D at the same time. And so it's the one that's on the surface of all B cells when they come out of the bone marrow. D is on there as well, but it's not playing a significant role. When they go through the differentiation process, the first antibodies that the plasma cells make are IgM. So when we talk about that lag phase, if there's any antibodies made during that lag phase and then that primary response, mostly IgM. It's not until you get into the secondary response that you start switching to the other isotypes, like the IgGs that are going to do a lot more. So IgGs are really good at neutralizing toxins and things like that. Tetanus toxin, and you're going to have IgG that's sopping it up, binding it up so that it can't do anything, can't bind to its receptor, and those type of things. So, anything else? Well, we'll have some time at the end to kind of reminisce on the day, and then we'll go through the whole anatomy of immune response to, hopefully we'll go through a bacterial infection, like a gram-negative bacterial infection, and also have time to go through what's going on during a viral infection, because they're fundamentally different, and how they start and how they proceed. But T cells don't recognize antigens the same way B cells do. Keep in mind, I think I only mentioned this once, and I was telling one of the students that if I say something more than once, like some of the things I've said about 5 times, those are the important things. T cells don't make a secreted specific protein, like B cells. B cells have an immunoglobulin on the surface to recognize the antigen, and then they go through the differentiation to make antibodies, secreted versions, so the secreted antibody can go to wherever it needs to go, wherever the site of infection is, wherever the toxin is, whatever it is that it needs to go bind to. T cells don't produce something like that. They have the T cell receptor on the surface. They physically have to go to where the action is, so that they can interact with the antigen-presenting cells that are present there, give them cytokines to activate them, activate the other effector cells that are in the nearby environment. So what we're going to talk about the next few hours here is just how do T cells recognize antigens, how do they get activated, and what do they do once they get activated. And so this slide is just showing you a cell that has, you have your nucleus, you have your ER, where we're going to have some proteins made on that rough ER. We're going to have the Golgi apparatus, which is involved in secretion. And it shows you an endosome that was just formed on the side. So you have, in some cases, we have phagocytosis of pathogens or particulates or whatever. Sometimes it's endocytosis, pinocytosis. We're just bringing a little bit in, we're bringing in a lot. And they form these vesicles inside the cell. And in some cases, you'll have lysosomes that can fuse and degrade, kill what's in there, degrade it down, and we'll have peptides in there. In other cases, we already have something inside the cell, and the protein's being made in the cell. So what I want you to consider is kind of an exogenous process and an endogenous process. Exogenous, something is outside, we're bringing in and we're degrading it, and it's going to be the antigen that we work with. Endogenous would be like viruses can't replicate themselves. They have to be inside our cells and use our machinery. So what they do is they replicate their nucleic acid, they replicate their viral proteins, and so they're using our machinery, so it's endogenous, it's going on. We have certain things, when proteins get made, sometimes they're faulty, sometimes they just get old, and we have this machinery called proteasomes that break them down, so that we get smaller pieces that can be degraded. And what happens in the endogenous pathway that we're going to talk about, so proteasomes are going to break down the endogenously made proteins. That would be viral proteins and our own proteins, cell proteins. So we're going to look at two different pathways. This is showing you what's going to be involved in the MHC antigen processing process, and the loading onto the MHC molecules that is important for T cell recognition. So in this case, you have a protein antigen in there, and this is a very simplified version, just so that you can get the idea that globular protein that we talked about, or globular molecule, that antibodies recognize and the B cell antigen receptor recognizes, is not recognized by T cells. It has to be broken down, and in this case, it is a protein. Just take my word for it. It's not a carbohydrate. Antibodies can recognize carbohydrate. B cell receptors can recognize carbohydrates. They can recognize lipids. They can recognize nucleic acids. T cells cannot. We'll just say that. There's always exceptions, of course. But we're going to just say that T cells recognize peptides, and they recognize it in context of self molecules called major histocompatibility complex, class 1 and class 2. And these peptides are actually going to be, you could actually have the same peptide on a class 1 versus a class 2, but they don't fit properly on class 1 as they do class 2. And so sometimes you have smaller, shorter peptides on the class 1 versus the class 2. But they work fundamentally the same way. They're going to look a little bit different. But there you go. So here's what they look like. And so let's focus on the one on the left. This is MHC class 1 molecule, and I'm calling it's just called MHC class 1 because we're really talking about all mammalian histocompatibility complex antigens here. Mouse, it's called MHC. Human, it's called human leukocyte antigen, HLA. So this is just a general view of what it looks like. It's a 2-chain molecule. The yellow part is the alpha chain, and it has 3 domains to it. It has the alpha 1, alpha 2, and alpha 3. And then it also has this beta 2 microglobulin. It's a separate protein that is non-variable. It is constant from all MHC class 1 molecules to the next one person to the next. It's going to be non-polymorphic. But the top part of this MHC class 1 molecule, it almost looks like two ducks that are cheek to cheek. That's forms that groove up there. That is the variable part, and it forms the groove. I told you, I'm going to show you a picture in a little bit. It almost can look like the hot dog bun, and then the peptide is the wiener that you put in there to make your hot dog. So that's a class 1 molecule, and then the bottom just shows you, it's the biochemistry. We like to show what these things look like. And you have some structures there, but the groove is kind of made up of 2 alpha helical structures. The key difference between class 1 and class 2 is that class 1, just one molecule, just one of the chains, the alpha chain, forms the peptide binding groove. In class 2, on the right side, we have a 2-chain molecule, and both of them are required to form the groove that the peptide fits into, an alpha chain and a beta chain. And again, the bottom parts of those molecules, both the yellow and the green, are going to be highly conserved. The top part is going to be the difference, the peptide binding groove, because they're going to bind to different peptides, and we want to make sure that we cover as many peptides as we possibly can to respond to. Let's take a look at what this looks like in the human system. The top panel shows you all of the class 1 isotopes. So these are HLA, MHC class 1 in human. And what I want you to notice here is there's 6 of them up there, but there's only 3 that really are of consequence to us. A, B, and C. HLA-A, HLA-B, HLA-C. Those are the class 1 molecules that will present antigen to CD8 T cells. This is going to keep coming up. Class 1, the CD8 T cells recognize. Class 2, the CD4 T cells recognize. The other ones you see there, E, F, and G, they play roles in getting the other ones loaded with peptide. They do different things that we don't really care about. We're not going to get into the details of those. What I will want you to notice is that they're colored. They're not colored just to be, pretty. They're colored by how many forms there are. So the red means it's highly polymorphic. Lots of forms of HLA, A, B, and C, the A chain, the alpha chain. Again, the beta-2 microglobulin doesn't change. There's only one form, so it's yellow. But the HLA A, B, and C, that alpha chain, has lots of different versions of it. The bottom part is our class 2 haplotypes, isotopes. And the ones that we're interested here, there's only 5 of them. Ones we're interested here are the last 3, DP, DQ, and DR. DM and DO also play roles in loading and getting the other ones up to the surface. It's the DP, DQ, and DR that are involved in presenting antigen to CD4 positive T cells. And you can see the polymorphism there. Both the alpha chain and the beta chain from DP and DQ are very polymorphic, so they're green, whereas the alpha chain from DR just has a few versions, and the beta chain has multiple versions. It's highly polymorphic. So what you need to be concerned with is the class 1s. There are 3 highly polymorphic isotypes that can be on your surface, and in the class 2 there are 3 not quite as polymorphic as the class 1, but because you actually have 2 chains involved, there is a lot of different forms of those as well. That's 6 MHC molecules that are on the surface of your cells. But the class 1s, as we're going to find, the class 1s are found on almost every cell. The class 2 are going to be found on specialized cells, like antigen-presenting cells. But I wanted you to see that first before we went to look at the actual numbers. So keep this in your mind, and let's look at class 1, HLA, A, B, and C. In the human population, they've gone and they've sequenced a lot of class 1 molecules, a lot of different people. And this is what they've come up with. That alpha chain for HLA-A, they've identified 218 distinct versions of it in the human population. The alpha chain of HLA-B, 439. And the HLA-C, 96. Now, I just told you that class 1 is going to be found on almost every one of your cells. It's going to be found on every nucleated cell. But you're going to have, you should have a version of all three of those, but you also have a version from each parent. So you'll have potentially six different MHC class 1, 2 out of the A, 2 out of the B, and 2 out of the C. Now it's quite possible that you end up with HLA-B 231 twice because you got it from both parents. But the likelihood of that is pretty low. You're going to have 2 out of 218 of the A, 2 out of 439 of the B, and 2 out of 96 of the C on your nucleated cells of the class 1. So a lot of polymorphism, and if you start to think about the math that's involved, the likelihood of anybody in here having the same 2 A, B's, and C's on their cells is very unlikely. And this is where we'll get into graft rejection and transplantation problems a little bit later. Here's the HLA class 2, and we're going to ignore the top 2 and start with DPA1. DPA alpha 1. It is there's 12 versions of that. The beta chain of that, there's 88. DQ alpha 1 is going to be 17, and beta 1 is going to be 42. The DRA and DRB, so that the DR class 2 is a little more difficult. It's a little more complex, and I'm not even going to go into it, but you only have 2 versions of the RA, but you've got these other versions of the B that you can kind of jump around to. It could be 1 of the 269 of B1 or of the 30 of the B3, so the polymorphism is really quite extraordinary there as well. Yes?
Do you want us to be able to recall any of these?
Oh gosh, no. I want you to understand that there are potentially six different class 1 molecules, and they're so polymorphic that the likelihood of you ever running into somebody with the same 6 is very unlikely. There are 6 class 2 that are highly polymorphic, and you are not likely to find those 2. Now, when you consider all 12 of them together, the likelihood goes up into the hundreds of millions, probably. Yes?
If you have a patient that has 2 of the same kind, are they at risk, more at a high risk for infection or immunodeficiency? That's
a good question, and it's probably not going to be at the level of just 2 of them the same. So here's the thing. There are things called haplotypes, and some of these haplotypes are the combinations that come together. There are some people that have certain combinations, like they have this DP A and B, and they have this DRA and B, and they have a certain haplotype. And if you start looking at some of the autoimmune diseases, some of the patients with autoimmune diseases, you start to see that there are certain haplotypes associated with autoimmune diseases. Now, the whole purpose of this is survival of the species. We need to be able to react. Somebody in the population needs to be able to respond to every possible pathogen that they come in contact with. And so sometimes it's just the luck of the draw. Some of the haplotypes, you end up with the class 1 and the class 2 genes that allow you to respond to a certain pathogen better than somebody else. That's a possibility. Some people will not be able to respond at all because they don't have a class 1 molecule. Let's just say a virus, influenza virus. This individual is missing class 1 genes that are able to bind any peptide from influenza, which would be extraordinary because there's lots of influenza proteins that could be peptides that could fit there. But if you can't bind to it, you don't have a T-cell response, a cytotoxic T-cell response. You will probably die from flu because you do not have the ability to respond appropriately. You have a hole in your repertoire. So it gets really complex when you start thinking about mixtures of these things, but there certainly is it looks like the people that have certain genes are susceptible to certain things, like multiple sclerosis, other autoimmune diseases. You go to OMRF, and there are whole labs that's what they work on. They go and find people that have certain haplotypes, and they want to study them, or they have a certain disease, and they want to sequence them and see what haplotype they have. So, bottom line, survival of the species, much polymorphism here so that somebody is able to respond to every pathogen. And I was talking after class a while ago. There are some people, when somebody gets infected with HIV and they want to determine whether they have HIV, the test that they give is a test for an antibody to HIV. But as we're going to find, viruses infect cells. They get inside the cell, and what you want to do is you want to kill the cell, you want to get rid of the virus. Antibodies can't do anything about that. They can't get inside the cell and do anything to neutralize the virus unless it's in there. So what you want viruses to be, you want antibodies against the virus to be around to sop them up if they're loose. But you need something to kill that cell, that host cell, and so the viruses can't replicate there. That's what the cytotoxic T cells do. We diagnose exposure to HIV by identifying antibodies to HIV proteins. It turns out that some people don't really make antibodies to that. They develop a cytotoxic T cell response and they clear the infection and they're immune. So there is a population of people that, because of their haplotype, respond with a cytotoxic T cell response instead of an antibody type response, and they clear HIV and are immune to reinfection. So, survival of the species. That's just one little example. So, any other questions about that? This is all good, because I want you to understand that there's a reason for this, but then there's consequences later. In modern medicine, there's consequences because of the ability for us to surgically move organs, to transplant organs, and then we have these problems that come up with it. Okay, this is kind of what I was getting at next, and that is two things. Polymorphism, so lots of forms, and you saw all the different numbers there. So there's potentially six different class 1 molecules on your cells, and potentially 6 different class 2 molecules on certain cells. And of course, they'll be on both, on these antigen, both of them will be on the antigen presenting cells. So polymorphism, because of the different versions. Polygeny, in that there's more, there's multiple versions of it. This is just showing 3, and you know that there's potentially, this is showing 2 alleles of 3, and that would be like a class 1, so it shows that you could have 6 if you had different alleles at on both of the chromosomes. So instead of, we'd have red, yellow, blue at the top, maybe on the bottom the allele is going to be orange, green, and purple. And so you'd have 6 different things on the outside in this multiple gene type thing. But you mix it all together and you really have what you see on the bottom, with the red, yellow, purple, whatever that is, blue, orange, green. So you have 6 different versions in like just the class 1 molecules. That's the most likely things that happen. It's possible you could have 2 of the same one, but not that likely. So polymorphism and polygenes make it very polymorphic type of population. This is just showing you what happens in case of one of these 12 that we talked, one of these 6 that we talked about. So 3 HLA class 1, 3 HLA class 2. This is just one of the genes. So the sperm donor is going to have 2 alleles, a blue and a red. The egg donor is going to have 2 in a yellow and a green. And there could be, they're showing 4 progeny that come out of this. And in one case, the blue from the male gets with the green from the female in the first case. And then we have the red mixing with the yellow. You can see that. In this case, you could have 4 different haplotypes for that one gene among the siblings. What you always hear about if somebody just needs to give a kidney, go to a sibling. A sibling might be the closest thing that you can find, and that's true. Because you got 50-50 chance that you're going to get this one, 50-50 chance you're going to go down the line. But in this case, it's quite possible, just with one gene, 4 different haplotypes. But half of them are going to match what what came from mom, and half's going to match from what came from father and yourself. But the siblings aren't going to be able to really get along that well. There you go. Let's move on to what these things do. So this is the hot dog butt. You see this, we're looking down on top now of the MHC molecule, and those there you can see this alpha helical structure from the alpha 1 on the left side on the top, and then the alpha 2 on the bottom. Remember, class 1 has one alpha chain that forms the whole groove. And so the alpha 1 and the alpha 2 form this binding group, and that's a peptide that you see running through there. I don't know if I'll be able to point this out, but let's see. See these blue right here, and blue, these light blue things there? Those are weak interactions between amino acid residues, amino acid moieties, some of these R groups or whatever that's sticking out of an amino acid, with amino acids in the groove. So that's a peptide going down through there in the middle that you see there. Those are hydrogen bonds. You know, I know you all have had your chemistry and you know hydrogen bonds. There's lots of weak interactions taking place, not covalent, but these weak hydrogen bonding, and there could be some hydrophobic, but not to the extent that we would think about. These are going to be hydrogen bonding type of interactions between the amino acids of the peptide that has been loaded in there and the peptides that are already in the MHC class 1. Basically the same thing going on in class 2, except that you have two different chains that come together to form the groove, and you have a peptide that hooks down in there. So that is what it looks like from the top to try to understand what is the T cell recognizing when it comes there. It's actually looking, it's going to recognize the alpha helical structures of the MHC molecule and the peptide that's down the middle. It's recognizing both. Got to keep that in mind. It's recognizing both the self molecule and the peptide that's in there. This just kind of gets at the differences in amino acids, because we've seen this with the antibodies. I wanted you to get some idea that there's variability between different types of, if you were to go in and sequence the different HLA molecules that we were talking about a while ago, you can see these places where there seems to be a lot more variability than others. Those tend to be the places where the peptide's making interactions with the groove. So that's where all the variability, most of the variability is going to be, is in those touch points for the peptides and how they interact with the amino acids of the class 1 molecule. All right, what's next? Where do you find these? MHC class 1 is going to be found as far as the lymphoid tissues, see T cells, B cells, macrophages, everything. So it's found on just about everything. Other nucleated cells, it's still going to be on that hepatocytes, kidney, brain, but it's not going to be found on the non-nucleated cells like red blood cells. So it's going to be found on just about any cell that viruses can infect, because it plays a role in recognizing that as a virus-infected cell. Class 2, skip T for now, and go to B. B's have quite a bit on them. Macrophages have some. Antigen-presenting cells like dendritic cells have a lot on them. Those are your top 3 antigen-presenting cells: macrophages, B cells, and dendritic cells. You also see them on epithelial cells of the thymus, and the reason why is because when we go through making a T cell, we didn't really get into the details about B cell maturation in the bone marrow, but you remember there was that positive selection piece where I said they had to make sure they had a functional heavy chain, and then they had to make sure that we got rid of the bad, the self-reactive. That's by touching other cells in the environment. The thymus is where the T cells are going to get educated. It's T cell school. And so the epithelial cells in the thymus also express class 2 because it's part of the education of the CD4 T cells. So we have quite a bit in there as well. T cells, you see a plus with a little thing there. Activated T cells can put a little bit of class 2 up on their surface, but they're not really going to play a role as an antigen-presenting cell. B cells can, and I'll explain how they do that later. They don't have good, they're small. Remember, they're a small lymphocyte. They don't have phagocytic properties. They don't have really, they can't do a lot of endocytosis. Well, how how do they bring in antigens? They actually do it through a B cell receptor. So they're more specific antigen presenting cell, and we'll talk about that later. Neutrophils, no. Hepatocytes, no. Kidney, no. Red blood cells, no. And then there's a little something on the brain. There are specialized cells in the brain called microglial cells that are very much like macrophages. They're resident up there, and they can put a little bit of class 2 up on their surface. So, bottom line here is just about every cell, every nucleated cell has some level of class 1 on their surface. But only specialized cells are going to have the class 2 on their surface. And that'll become clear as we go through this. Where do you, what role are they playing? Okay, so let's talk about a pathogen that somehow gets into the cytosol, like a virus. Viruses have evolved to bind to certain receptors and get inside our cells, and then they take over and use our machinery to make more of their virus babies. What happens to them is the proteins, and I mentioned the proteasome, the peptides that come from the transcription of the viral genes will end up being degraded by the proteasome in the cytosol and loaded onto class 1. Put up on the surface of that cell and basically say, looky here, I'm a virus-infected cell. Where is the CD4 or the CD8 cytotoxic T cell that is specific for me? Come and do your job. And they do. They come and they kill. They induce apoptotic state in the cells and they die. Intravesicular pathogens are going to be like the macrophage gobbles up a bacterial cell, is one example. And remember that phagosome is going to bind to the lysosome, and we're going to throw a lot of stuff at it, including a low pH and some really bad enzymes, and we're going to kill that pathogen. And what comes out of it is some proteins that get degraded, and those hook up with the MHC class 2 molecules in the inside the cell and are transported up to the surface of the macrophage. And there, what they're going to do is present that to that activated T cell that got activated by the dendritic cells back in the lymph node and has now come to the site of infection. And so we're going to stimulate that T cell again, and it's going to start producing lots of things to kill. In this case, it would be like gamma interferon. The T cell would produce gamma interferon, activate the macrophage, and the macrophage would kill the intracellular bacteria or parasite, whatever's in there. Finally, we have toxins or something like that, small things that a B cell could bring in, and they bring it in through the B cell receptor. And so, in this case, the other two cases, they are very non-specific. A virus is only specific in that the cell that it infects has to have a receptor for it. The other ones, the macrophages, they'll gobble up anything. They don't care. But in the final case with B cells, the machinery to bring something in to process and put in context of any of these molecules, it has to use its B cell antigen receptor, which is specific for a certain antigen. So if it's specific for the tetanus toxin, then it can bind to the tetanus toxin, bring it in, process it, and put it up in context of class 2 molecules and present it to T cells that are specific for the tetanus toxin. Now, this is great. It's perfect, especially if you want these B cells to make more antibodies to neutralize. This would be a fantastic thing to do. The problem is, in primary responses, remember, we don't have enough of these B cells around. They're still naive, the T cells are still naive, and so we have to vaccinate and get the numbers of these cells up, get the affinity for the antigen up, and get the T cells going before this really plays a significant role. But it is a great way to present antigens to T cells if you're in more of a secondary immune response. This next stuff is just kind of giving you a little deeper detail on what's going on. You're going to see some figures that have a lot of protein molecules in there that we're not going to get into. I just want you to appreciate what happens with these peptides, how they get loaded on the MHC molecules and then go to the surface. In this case, we have the cytosolic produced peptides. So we'll just call it a virus in this case. The virus is growing in the cell. transcribing its genes, and then that's being translated into a protein. Some of those proteins go to the proteasome and get broken down into peptide fragments. Your cell molecules are doing the same thing. Proteins inside your cell that get old or whatever, or quit working, they get degraded by the proteasome too. And they're going to have the same thing going on. So those peptide fragments could be a viral peptide or they could be a cell peptide. They get moved into the endoplasmic reticulum through TAP. And so TAP is this transporter that moves peptide fragments from the cytosol into the endoplasmic reticulum, because that's where it's going to run into MHC class 1 molecules in the ER. And this is where it's going to show you. If you look over to the left, you can see the MHC class 1 molecule, the alpha chain, and you see the groove, and you can see that beta-2 microglobulin is not really associated with it. This is not a functional class 1 molecule at this point. These other proteins, we'll just call them chaperones because I don't want to get into what's going on there. But the purpose of all these other proteins is simply to prevent anything getting inside that groove until we bring some peptides into there. We don't want just anything falling in there. And so, in fact, what's going to happen is the peptides get degraded by the proteasome, they move through TAP, the process a little bit more, and they are tested to see if they fit in the groove. Some will fit, some won't. And I'll tell you a little bit later that it's estimated that each of these molecules can hold up to 10,000 different peptides. So some of them are going to be very low affinity, some of them are going to be much higher affinity because of the touch points where these weak interactions take place. Multiple weak interactions hold the peptide in the groove. The more and stronger the interactions are, the tighter the binding is going to be. So peptide gets put in the groove, beta-2 microglobulin, and all things comes together, and then it pinches off from the ER in a vesicle and then goes to the surface and put on the surface. Class 1 molecule is now on the surface with a peptide that it got from the cytosol. Whether it's self or virus is just, we'll just wait and see. Same thing is shown on the right side versus the left side here. The right side is showing that same thing, but it's here you've got this intracellular antigen, proteasome degrades it into a peptide. Those peptides transported into ER. They get loaded on class 1, and it's really showing you the processing through the Golgi and then into a vesicle that is then moved to the surface, and you have MHC class 1 presenting the peptide at the cell surface. That's the endogenous pathway. The exogenous pathway is the one that I told you about, where you see on the left side, MHC class 1, we have some kind of extracellular antigen, like a bacteria, that is being engulfed. And so it's in this case, endocytic vesicle, where it's being degraded and all the way down to peptides. And so we have this vesicle that has in it, besides a lot of things, membranes and anything else left over from the degraded pathogen, but there are some peptides. And this peptide, this vesicle, then binds with something that's holding the class 2. And what you're seeing here is a class 2 MHC molecule is forming in the ER. It's then transported through the Golgi, and then it moves up and fuses with the peptide containing phagolysis. And peptide, as it fits on there, you're going to see this in a little more detail on the next slide, goes to the surface, and now you have class 2. So MHC class 2 is an exogenous pathway. MHC class 1 is more of an endogenous pathway that involves proteasome. And this is just going into a little more detail so you can visualize it, but here's your, this is just a protein showing something, a globular protein being taken up by pinocytosis or endocytosis. And then it is being degraded in the third panel into these peptides. Along comes a vesicle containing the MHC class 2 molecule that fuse, and if the peptide fits, it gets in there and it binds. If it doesn't, another one is tried until one of them is found that can bind it. This is showing you where the MHC comes from, and this is, again, there's some additional molecules here, the invariant chain that is basically there to hold the groove closed so nothing else could get into it. You can see this green hook-shaped thing. That is just setting in there as a kind of a chaperone to make sure that no peptide gets in there until these exogenous peptides are added to the groove, and then it's taken to the surface and expressed on the surface. There you go. That's the same thing we just saw, just showing us two different pathways. That is basically the MHC and how it's processed to be presented to T cells. So, I think next we were just going to move on to, we'll get started and then take a little break on the next one, which is T cell maturation. It's really kind of getting into